I want to introduce you to Dr. Jay Evans. He's a research, not a leader anymore, research entomologist at the USDA-ARS in Beltsville, Maryland. It's really hard to pinpoint one thing you do because I think every time I read a cool paper, you're involved with it somehow. But I guess in general, you develop molecular techniques to manage pest pathogens and diseases for not only current issues for honey beekeepers, but also emergent issues such as trophoid lapse and things like that. So I'm not sure if you can touch on that, but I'll let Jay kind of take it away from here.
All right. Well, thanks, Garrett, and thanks for the chance to talk. I have known people at Purdue for a couple of decades at least with Greg Hunt and Crispin, and of course, I'm glad you recruited Brock, whom I met as an undergraduate in Canada once at a seminar in the university that wisely changed its name to match his. But he's been a star for a long time, and I can tell with students and the support there, he's taken off and doing some really cool stuff. So look forward to working with your bee people over the years. So I will start. If it wasn't inherently obvious, I am in a fairly applied laboratory at the USDA. It's a honeybee lab. We do get to dabble in some other insects, especially in my case, with genetics and genomics. That's been really fun. But we are here and funded to tackle disease issues in honeybees. So that's what I'd like to talk about today. And many of you, or many at Purdue at least, are beekeepers. And I think there might be some beekeepers also in the group, in the audience from Indiana, which has an active group. And so you get the gist that honeybees live largely in artificial boxes that we provide. They build their colonies around a framework that we provide. But in the context of disease, what's important is that each of those boxes has tens of thousands of individuals, and all of them at some point are susceptible to disease. So there are brood diseases in the larvae and the developing bees, diseases of queens, workers, and the male drone. So there are multiple risks, I guess, for biotic threats. And since they're so crowded and living within these colonies, they are pretty good at sharing disease once it gets going. And I'll talk a bit about that in terms of our virus work later. Commercially, bees are, of course, very important for agriculture, and they're moved all over the country. And that brings with it the potential for spread of disease, and at some level, some stresses just from the movement and the changes of the environment. And we're still not sure as researchers or as extension bee people and just in the community why this is happening, but perhaps as a result of those stresses, honeybee losses have had a bit of an uptick, and they've gotten a lot of attention, I will say too. There weren't these sort of national surveys at this scale until the mid-2000s with colony collapse disorder, but they have revealed some really high overturn of colonies. The net number of honeybee colonies is holding steady thanks to the economics of it all and the passion of hobby beekeepers, but the loss rate that they're facing in terms of colonies is in some years approaching 50%, 40 to 50% annually. So that's a big problem, and it's an embarrassment for us as researchers because the beekeepers really want it back down to where it was, which is maybe 15%. These losses are pretty consistent nationwide. This is 1 year total. The loss rates shift a little bit, but I will say the upper Midwest is consistently pretty high for honeybee losses. In that sort of 40 to 50% range annually. This is a neat review paper from Italy, actually, and I like to start with it because I think they kind of very clearly lay out, one, that we're not sure what's going on in terms of these actual losses as scientists, which is always unfortunate, but we do have a good feel for which factors combine to be most impactful. They focus on three of those: nutrition, an agrochemical threat, and then the sort of disease angle that we think is really ultimately most important. But they are interactive. chemical stress, not necessarily of neonicotinoids, but others as well, can impact physiology and immunity. And of course, poor nutrition, as with us, can do the same. But what really starts to happen is these mites, Varroa, and their, especially one species or quasi-species of viruses, really have had a consistent impact on honeybees individually and as a colony. And this too, and this is a chemical stress related paper that a USDA colleague, William Meikle, was involved with. And I will say this story that I'll talk about is almost identical, we think, for the viruses that we study, where if you follow individual bees through their lifetimes, and these are female worker bees, you will not see this big pile up of bees outside of a colony or in an apiary or on the street, in a parking lot, but you'll see a consistent decline of worker bees as time goes by. And if there's a chemical stress, and these are bees exposed at a very young age to a chemical, the cumulative effects of that on time spent foraging, which is money for bees because they're bringing back food, and the numbers of flights, it adds up. There are some superstar bees here that are foraging a lot, despite the same chemical exposure, but on average, bees are living less time and doing less work. And this is shown here as well. There's just more of these kind of red superstar bees, as it were, in the control groups than in those exposed to a chemical. And that's the same with disease. And the net effect, if you look at it from survivorship, is just a little steady drip of workers that's a little faster when they're exposed to stress. And this is exactly what we saw with virus stress in bees, that basically middle-aged bees are not doing great. And when that starts to happen, especially in the winter or in a time when you need bees to hang in there, the colony will collapse over time. They'll start by not getting as much food back into the colony, and then eventually they just can't, the queen can't keep up. On the disease side, this is caused by a whole suite of things, and it looks like human disease in many ways. RNA viruses, a few DNA viruses, but not much. They're mostly on the RNA side. Some bacterial disease that's important for brood and at least one for adults. Fungi, protozoa, including a protist group that I'll talk about. And then over here, these mites that have a physiological effect when they bite into bees, but they also are pretty good at moving viruses at least around. So a lot of our work, being in an applied setting, is to deal with things after the fact. And we go out and collect from die-offs of bees. This happened to be from the colony collapse die-offs in 2007, early spring of 2007, when we started collecting. Take them back to the lab, and not knowing what to look for, we mash them up. This is an RNA extraction of 50 bees, which is kind of a blood sample for a colony. And we've done a number of things, from RNA sequencing to just straight up qPCR, to try to sort out the microbes in the bees that have died versus those that are healthy and surviving. And it was unsatisfying a little bit at the start that we thought we'd find the one virus. Many times there was the one virus in one study that jumped up, and people said, aha, that's the cause, or a disease that was the cause. But what really looked to be happening is a number of pathogens and parasites increased in these sort of so-called healthy colonies and the ones that were collapsing. This DWV is deformed wing virus, and that's really become one of our focal points. Myself and Dr. Judy Chen in our lab are pretty convinced that that's causing, or at least it's a possibly addressable cause of a lot of bee problems. It's named because if you inject bees, or if a mite injects bees with a really high titer at the right time, they will develop without expanding their wings. So they have these sort of nubs of wings. And that's rarely seen, though. That's when that happens, that's really the end of the road for that colony. But instead, at lower doses, they get that sort of steady decline, especially of kind of middle-aged bees when they start to forage or get a bit older. And there's some learning differences in bees that are impacted by this virus and various things. It is completely widespread. These are just different years, different incidence rates of these. It's in virtually every colony in the US, and it's probably impactful in most apiaries, especially if the mite levels are pretty high. We've done a bit of work lately just trying to sort out why it's not always deadly, right? So you think, okay, here's a bad virus. We can't get rid of it. The bees have it. A lot of bees seem to do okay with it, or at least there are bees that survive fine. This is work by Dr. Eugene Ryabov, who's been a visiting scientist in the lab, basically sorting out using infectious clones. So these are native strains or variants of this virus from our area, but they're a little bit different. And he was able to synthesize cDNA clones for these, so we could actually do experiments, kind of the first experiments directly tracing defined strains or variants in beehives. For better or for worse, they all did equally well. These are kind of growth curves over time of the 5 that we initially started with, about the same as the wild type virus that we were able to measure. But what's really this has turned into, it's just sort of interesting, is there is a sort of, we think, a balancing selection or a negatively frequency dependent selection for slightly different strains, not vastly different in this study, but slightly different strains. And that might be because of the bee defenses relying on RNA interference. We think that could target the primary variant, and there is a way through rapid mutation, which these viruses are famous for, to possibly escape that and have rare variants do better. One rare variant that's done really, really well, unfortunately, is at times called Varroa destructor virus, but it's also called deforming virus B. It's a variant, it's a subspecies, as it were, of the same species. It popped up in the US around 2010, and this is from 2016. It was already equally prevalent to the old flavor of deforming virus. And it's now slightly higher in frequency, and that rapid sweep, it's sort of echoed by diversity of this virus. Just to focus here, the blue line is the nucleotide diversity of isolates from this virus in the modern day, actually. And then this is for the old strain. And so it still hasn't picked up this vast amount of diversity that the old strain has, but it's done exceedingly well. So it's clearly somewhat better, at least, at infecting bees and succeeding as a virus. And the reason we think that's happening is because of mites. So these parasitic mites, it seems this variant of deforming virus seems really, really adapted, or at least tolerant of being hitchhiking along with mites. So I'm going to talk about the mite story, but I want to say just one other interaction that has long been an interest of mine and is, we think, important again for viruses as well as other disease, is that, this is an individual bee is the unit that gets sick with the virus, of course, and she will live or die perhaps with a virus infection, but she's surrounded by sisters who feed her and take care of her. So I'll talk about later, they also share or receive diseases that way. And then a whole suite of other colony members who keep hygiene in the colony. I know your group is doing really active work with the mite biters and with these hygienic traits that are hugely important for colony health, because they keep diseases at check. And then outside that, of course, there's a little bit of patrolling where they just keep other bees most of the time from coming into the hive and thereby bringing disease. But it's really the individual bee that's critical, and especially with this mite story, if you are parasitized by this mite, Varroa destructor, your virus titers go up a couple orders of magnitude often. They really, really go up. The mites seem very often infected with the virus, or carrying the virus at least, and bees that get bitten, because of the nature of the sort of chewing nature of the mite, the virus is inevitably injected into the fat cells actually in the fat body, as well as the hemolymph and hemocytes. The mite itself is worldwide except for Australia and causing big problems. So we have a large group. I'd say Dr. Stephen Cook is the lead of this group now in terms of practical outcomes of mite research, looking at mites. It's a bandwagon that many of us have jumped onto because we think it's important for bee health. In terms of viruses, we can follow the viruses and the mites in the lab now, because of techniques for rearing mites and keeping them going long term. We just had a female mite that lasted 75 days in the lab with successive bee pupae as her food source. And these sorts of experiments, kind of combined with tracking viruses, have helped us figure out a bit more about how the virus is tied to the mite, and then maybe how the mite and the virus combine to impact bee health. And this is one study that's just come out. It's a look at using labeled virus, so we know that virus over time. We know it's not one that was just sort of latent and popped up. Getting mites infected with it, and then those mites are put upon clean pupae here. And honeybees are pretty good. If pupae are diseased or challenged in some way, they will actually cannibalize them, remove them from the hive, but in doing so, they'll actually chew them up. And this is just a confirmatory study to show that as they chew up these diseased pupae, that hygienic worker gets infected. And then she's quite good over time at sharing that virus infection with her nestmate. So it's another route. And what's worrying about this is this continues even if the mites are controlled, as beekeepers tend to do. So once the virus load gets to a certain level, that sharing by trophallaxis is a big problem. And it doesn't require much contact. We've done experiments through screens here where they're just touching tongues. And it's really, really efficient mechanism of kind of spreading virus across the colony. And of course, this happens in non-hygienic settings too, just whenever the bees are feeding each other. But we think this act of removing the sick bee leads to a super donor, a super infective worker bee here, and then she again then can be a source for many infection events. Another maybe more optimistic interaction that's had a lot of work lately is with the microbiome of bees. This is a bee on her birthday just popping out of her capping. And very early on, she will receive a dose of bacteria from her sisters or from grubbing around on the pollen, the nectar, the wax of the hive. And that dose of bacteria flourishes and becomes a pretty stable, pretty simple, but pretty stable community in the gut. And thanks to Nancy Moran's group, we have a really good view now of what comprises the bee microbiome, and then possible benefits for bee health, benefits and negatives, of course, for bee health. We got interested in this because we could see how a bacterial community would interact maybe with some of the parasites we were really interested in. And this study was with a parasite that's in the ileum, or it's quasi the small intestine, just the start of the hindgut of honeybees. It resides there and gets its nutrients from the bee there. That also happens to be where many of these gut bacteria form a kind of a biofilm, and so it was natural to think that those bacteria, either physically or in terms of interactive effects otherwise, could impact disease. We did a couple experiments. One is to take bees, inoculate them with a particular bacterial species that we thought was impactful, paint mark them, and put them back in the hive. So these bees, with or without the bacterium and with or without a parasite challenge, were dropped back in the hive. And then we go with a handy bug vac and pick up all the different paint colored bees at the end of it. And this was, it's good that we did this. The result wasn't as desired because we were thinking of ways that these bacteria might be co-opted to help bee health. And we'd seen actually on this bottom chart, the red dots are bees that had gotten the bacterium as well as this parasite Lotmaria, which is the trypanosomatid parasite of bees. In the lab, things look great. So, well, there's not a clearing of disease, but bees with this one bacterium had lower parasite loads. However, when we did these sort of mark recapture studies and put them in a more natural environment, the opposite happened. Bees, given the bacterium, this one bacterium at a young age as they emerged, actually were far more susceptible to the parasite later on, over a week as they developed outside in a colony. And the short story of that is that there's an interaction with another proteobacteria, Gilliamella, and the bacterial load where we basically engineered a kind of dysbiosis that did not impact bee health in the sort of sterile lab environment, and thereby kind of did protect the bees, we think, from parasites. But out in the real world, it threw things off so much that those bees were just kind of profoundly susceptible to disease. And so it's back to the drawing board for that. Nancy's group is continuing with some work to kind of work towards maybe pro or prebiotics for this, which could be neat, but it will have to be nuanced. The easy fix of this one species did not seem to work well. The other practical outcome, and this is largely from her group as well, is that antibiotic use by beekeepers disturbs these bacterial communities, and it could well lead to a greater risk of disease. This is from my colleague Judy Chen and her visiting scientists, showing exactly that for Nosema disease, which is a kind of persistent, it's a microsporidium, so a very basal fungus that causes disease in animals. In bees given, and this isn't the beekeeper or antibiotic that's used for say brood disease, but a pretty cleansing dose of antibiotics, so it's a pretty maybe severe case. But given that antibiotic treatment, they then were highly susceptible to this disease. It did not grow well in the absence of antibiotics. It grew incredibly well when bees' guts were cleaned of bacteria. And the bees did very poorly. They died when they had both the parasite and the antibiotic. They died very quickly, but just with the antibiotic and no disease treatment, they also had a foreshortened lifespan. So the antibiotic treatment, clearly showed that these things are doing something beneficial to bee health. If you're interested in the bigger picture of all this as students or that, and I'd encourage this book, which is a really fun read. It does have a little snippet on some of Nancy's work in it, but it's mostly focused on human microbiome studies and stuff like this, which is that we are moving toward an age of medicinal uses of maybe reinstituting or regrouping directly our microbiome. This isn't just yogurt bacteria and things like that. This is human bacteria that are transplanted, as it were, into new patients, and it's worked very well in some cases, and it's worked poorly in others, unfortunately. But I think that's where we are with the bees as well. Is these sort of probiotic treatments look promising in some cases, and then also look like we still need to learn a bit more on making sure they're safe. So now I'd like to talk about a few projects that have popped up in our group broadly over medicines, because that's really one of our main mandates, is to look for novel treatments for beekeepers, either to get away from the two widely used antibiotics or to treat disease that we don't yet have a real product for. And Judy's lab has done a lot of this stuff and had some really intriguing results. One is with the group of the cyclodextrins, which are basically a sugar polymer, I guess, or a sugar entity. Interesting in that they self-form as sort of a cavity or a cup, and that cup seems to bind to xenobiotics like pesticides, and whoops, sorry, quite surprisingly, and I didn't get the data slide in, so you have to look for this paper. They also reduced virus loads. So somehow this molecule that's sort of a mopper-upper of sorts inside the bees, presumably inside their guts before it's degraded, is able to sequester small molecules like some of the pesticides, but it also impacts virus disease. And so that's been really exciting. Further out there, we've done a little collaboration with Steve Shepard, a professor at Washington State, and Paul Stamets, who's a mycologist fanatic, on extracts from various mostly polypore mushrooms that are bioactive in other realms. And so they propose that these would be bioactive in bee disease. Sorry, this is a very hard slide to see on a laptop, but the short answer is in these sort of cup cage studies, there were some that were that result, then when the bees fed upon them, they resulted in lower virus loads. So there's a hint that that some of these compounds may do that. And this is a general set of, I guess, candidates in the realm of natural products or products that they're often synthesized in these days because of just replicability of getting them, but they're products that are in plants, that are in the natural environment. One practical reason to look at these is regulatory. They often have a shorter, less onerous regulatory pathway if they're can be shown to be already, say, in the human food stream and things like that. So we got into this as well because I'm not a chemist, but I did understand that nectar compounds had been proposed to be good for bees. So we started with that route with some great ecologist colleagues who did know a bit about plant chemistry. And some of the usual suspects in bee treatments looked promising. Thymol, which is used in one form as a miticide in bees already. Nicotine, which is obviously bioactive in good and bad ways for insects. And so this is a preliminary study, nothing great, nothing that cleared the deck of viruses, but looked promising. So we've gotten a grant now to look more broadly at this and to try to screen dozens or hundreds, hopefully, of compounds. And we're in the midst of that right now. This is just the flow chart. We have about 400 to go, so we're not that far along. COVID did not help, but we are making some progress, at least to screen through these. A diversity of things, very few of which I've actually come up with. It's been a team with a plant chemist and also just looking in the literature, I guess, looking for examples for antivirals from natural sources. We did ask the bees. I do like every once in a while to do a behavioral study. So we asked the bees if they would tell us, what to work on. And so this is just one of our feeding assays and these are basically just some of our candidates in water. Maybe it was like 5% sugar just to get the ball rolling. But the idea was to see if we could find compounds that they would move into that maybe they would so-called self-medicate or indicate that these were beneficial. We did get some preferences and followed up on some of those. I wouldn't say this is the route that led to any lead candidates at this point, but it was kind of fun while it lasted. What we've done instead is to just do it by brute force. So we set up a lot of cup studies with bees. These are maybe 30 bees individually. For the viruses, we have often just used older bees that we knock out with CO2 and then shake into these cups and then try to medicate away things they already have, because it's pretty hard to consistently inoculate bees with virus orally. For the gut parasites, of course, we can do the oral inoculation. That's what's going on here, where we take very young bees and give them a parasite. and then try to keep it from growing. And this is now seemed like a decade of this, but it's been at least 4 or 5 years of lots and lots of these cup studies. This is actually from just a couple weeks ago, and we're trying one with a high school student that, thank goodness, we finally can have exceptional students back in the lab some of the time. We're at 25% capacity, but we get some students in, and just sort of again testing in sugar water feeders the impacts of different would-be treatments. And so, yeah, we've done, now I think it's probably 60 of these by now, and we're still chugging along, hopefully into the fall, if not winter, as we do these older bees. There's a handful that look kind of promising. I'd say we're ready to unleash on the beekeepers or regulators or get partners on and stuff like that per se, but there are some promising ones that at least are changing virus levels. But we're trying to streamline this more, and especially be able to do something where we can do it without going in and out of beekeepers all the time, beehives all the time. And we're back to this virus test that invokes these sort of labeled viruses that are traceable over time, and that's proved to be, I think, cleaner way to do these assays in various ways, and it's a bit cheaper. It's maybe 1/10 the cost of our current genetic screens at this point. So, and I can talk later in the questions or elsewhere about the 2 flavors that we've been using, but to cut to the chase, the one that we favor right now for quantifying virus growth is it's a luciferase expressing virus, nanoluciferase. Tends to be a bit more stable in the virus genome, and tends to be pretty quantitative in terms of reporting back how many virus copies are there. And I love this project. It's very meditative to pull these pupae out of frames, and it's within my skill level. So I pull the pupae, and then my technician Dawn does the injections, and she's much better at that. So we pull pupae from beehives. We inject them with, say 10 to the 5th virus copies with this label. And it may or may not also be co-injected with one of these chemicals. And these are the types of data we get. They're surprisingly pretty clean, pretty tight with that injection scheme. There's still a lot of variation, of course, but from past studies, we think that's from bee to bee variation, probably genetic variation. So we're not too worried about that. This is actually an offshoot of this. We did an immune stimulant would-be treatment in the field, and I spent 2 months doing a field project. And this lesson for you grad students is to walk before you run, because although it was probably good for me to get outside, but it was a lot of work going along. And in the end, you could not picture a more tightly tied set of means for my treated ones here and control ones here. So there was zero effect of this after way too many checks on beehives and stuff. But it did validate, I think, the technique. These are actually pupae pulled from each of these individual colonies, injected, and then checked for their responsiveness to virus. And it pointed to something that's just intrinsic to bees. And we have a paper out with a beekeeper, Danny Weaver, just recently. It's about a probably 8 to 10-year-old study now, because it was very much backburnered by life. But fortunately, we finished it. And the same thing happened. He has some virus resistant or tolerant, at least, types of bees. And but within those, there's some bees that are really, really bad at keeping out virus up here, and some that are quite good. And and it varies colony to colony at a high level. So for those of you doing breeding, which I'm not a bee breeder, there is a ripe amount of variation for virus resistance out there. And it's just waiting, I think, to be tapped. I just wanted to highlight one other study from the lab. Also, we had a COVID arrival of a scientist, Evan Palmer Young, who I'd worked with before when he was in Lynn Adler's lab on these phytochemicals, but he was a bit orphaned during COVID, sadly, and of course we snatched him up. And his interest is in trypanosomatid parasites of bees, these sort of gut parasites, one of which we actually had named because it was anomalous to what it should have been at that time, in a new genus, Lotmaria. And he's really interested in kind of physiological impacts of these. He's also done, of course, a lot of neat stuff with nectar chemicals in these. He's really driving the nectar chemical stuff. But on the physiological side, there's sort of an interesting contrast. So these two red ones are two trypanosome parasites that have been found in honeybees. Crithidia mellificae was the historic one, well known, a couple of decades of work, kind of petered off in the 80s, not much done on it, for about 30 years, really. That parasite really, really tolerates high temperatures. These are, 35 degrees C, right in there for the hive temperature in the core of the brood box. Lotmaria, which is totally displaced it by now, is a bit cooler, but still pretty warm, especially warm compared to these mosquito trypanosomatids in the same, roughly the same group. The generic names of these are probably going to continue to flux a bit, but they're all a branch of the trypanosomatids is distinct from, say, leishmania and of course trypanosoma, which are the ones linked to human disease. The bumblebee parasites, temperature-wise, had some variation, but were kind of like this Lotmaria, not quite favoring the high temperatures that Crithidia likes. Then he did the same thing, but with pH. And again, this is a measure maybe of these gut microbes shifting the pH. And again, the two B ones were fairly different from each other. And so our gut feeling now is maybe Lotmaria is kind of working its way into the system from somewhere else. We don't know. It's probably not a mosquito, which are down here, because they actually look more like the historical bee one. But there's something different about these 2B parasites, and whether it leads to a way to control Lotmaria, I'm not sure, but it's been intriguing trying to sort out the physiological changes there. So I'm going to end there, so we have ample time for questions. But yeah, I do want obviously to highlight our group. There are 5 of us as sort of honeybee specialist scientists, and a great technical staff from beekeeping to genetic work. But half of the folks here were visitors. So from overseas, when that was a thing, hopefully it will be again in 6 months or 8 months. We've had a lot of great collaborators from overseas, including Eugene Ryabov, who's here and is a true virologist, really, really good virologist, and brought a lot to our group in that fashion. So I will end there, and I would be happy to entertain questions. I'll take the screen down, but if you want to see something, please tell me. Hopefully I wasn't muted for 45 minutes. Did that work? Okay.
Okay. Yeah, that was great talk. I'll ask one question. So in the beginning, you kind of talked about this kind of this frequency-dependent selection, in the viruses, and so for to form wing virus, for example, they have a host like the bee host and the mite host also. Do you think that's kind of dependent on these differences in hosts between the mites and the bees? Like, have you looked at the mite side of it, and if there's some coevolution between kind of the viruses and the mites?
Yeah, nobody, as far as I know, has looked at it in terms of RNAi in the mites, though they do have an active, they do that. What's interesting in that realm, so this DWVB, or Varroa destructor virus, which was first named in a study that found it in mites, seems to be way better at actually replicating in mites. In fact, we can't get to replicate in mites. The flavor of deforming virus A that's in North America seems to be passively transmitted, or sort of vectored as a stays alive pretty well in the mouth parts of mites, but it definitely doesn't go into their, well, it doesn't seem to go into their cells and actually replicate. And that was different than the lore for it, because it's clearly tied to mite levels. It clearly goes up when the mites are there, and the mites bite the bee, and there's big influx of virus soon thereafter. But it didn't seem to grow. It didn't seem to replicate. But the other one does seem possibly to do that. And so we think the main difference there is in strain evolution for which ones can actually replicate inside the mites, I guess. But none of these, we haven't done any of these subtleties of, this version of A outcompetes another, or there's RNAi. That would be neat to do, though. I think someone should do that.
Jay, I'm going to jump on as Garrett's scrolling through. I'm curious about Bee Reporter. So which strain are you using for it? And maybe you can elaborate just a little bit more on how it works.
That's a good point. Yeah, so it's one of those, it's a Maryland strain of deforming virus A. And so we purposefully, at least in our area, we're not using it outside or anything, but yeah, we didn't want to engineer or change the native strain. Eugene has labeled a deforming virus B, but unfortunately he hasn't tried with any of the fluorescent reporters that can kind of be seen in real time. Like GFP works great in it, and you can actually track it in pupae, put them on a light table, and see them. You can check every hour if you want, and that's been really neat. But we only have that one flavor for a non-lethal one. The luciferase needs to be exposed to a substrate to luminesce, and so we kept frying the bees to count that one. Although it can be a very crude, like that picture that I showed, Eugene's very fond of just kind of sticking a pipette tip in them and tearing them a little and dropping the substrate, and you will get fluorescence. But to quantify it, we've been putting them into a, almost like an RNA extraction, well, a very simple one with just basically PBS and some glass beads and macerating them, and then putting the substrate in, or adding that to the substrate, a fraction of that. So I guess we don't yet have that working in a sort of real-time thing for competition between strains, but that would be really fun to do. I think the key would be to get one of these, yeah, I guess it's what, ruby red, and I haven't done it. Yeah. It should not be hard. And again, I think maybe it's better to talk to Eugene about how these do have, of course, rare restriction enzyme sites along the clone that are engineered into it, so you could pop out GFP and then put in another one, I bet. So yeah,
that's really neat.
But yeah, we're hoping to share it. I mean, obviously the use of these are subject to biosafety committees at any institution. And there's no intent to use them in the field or that, but they are, if anything, less virulent than the native strain, because they have this, these viruses are very economical in their genome size, and they're hauling along another, in one case, I think almost 400 nucleotides, in the other it's maybe 500 and some. So they, yeah, they're safe, we think. So.
Yeah, really neat. Thank you.
Yep.
Okay, I'm not seeing any more questions. So yeah, with that, I guess, yeah, thanks for giving the talk today, and I really, really appreciate it. I've definitely learned a lot, so.
Okay, no, thanks you all. And yeah, send an email if you have questions or can't reach, Judy or Eugene or colleagues, and be happy to direct questions to them too.
Thanks again. Oh, hold on, one just popped up.
Okay.
Jay, can you? Oh, go ahead.
Yeah, okay. I'll just read it. Does Varroa also transmit deformed wing virus? Is there any work done to strengthen the bee gut microbiome to prevent the deformed wing virus transmission?
A little bit. So Nancy had a student with, again, with this, there's been a lot of focus on this one of the 8 core species, Snodgrassella, because it tends to be as closely adhered to the gut wall as any of them. And we did one study with and without that and found that if you injected the bees with virus, there was an improved, a lower virus amount when they'd been fed this one bacterium. So there's some suggestion that there's an indirect route where having the bacterium in the gut triggers something that makes them more likely to fend off a virus in the hemosyl, which is where the injection goes. Unfortunately, we thought, oh, then it should really work if you feed the virus. And again, feeding viruses to bees is not the best way to induce an infection, but you can do it. And as much as we tried, the bacterium in our hands did not slow down infection by feeding very much. But that's, yeah, it would be neat. And again, it may be a cocktail of, reconstituting an optimal gut community might be the way to do it if that were a treatment. But it's worth trying. I don't think anyone's come up with like one of the 8, say, that's really truly effective against viruses.
Okay, so we had a question in the chat from Steve Neff, and he kind of mentions so trying to breed for hygienics isn't necessarily a good idea. That was sort of his question. Can you talk more about kind of breeding and
someone else emailed about this, and I'm sure they've emailed Francisco and Eugene also. I don't think I would not go that far. I just think, hygienic removal of bees has to happen. And it's such a positive trait in other ways, right? Because it affects mite levels in the first place. So we did not do that experimentally for one. There was no test of, and none of this was at the colony level, for example. I think it's just pointing out a way that both the trophallaxis route to me is most interesting, that when a highly infected worker is in the hive, and she could get it from biting a pupa, but she could get it also by being that pupa that was bitten by a mite. And when she's in there, she's a super spreader with her sisters. But the net, I think there's been colony level evidence that the net impact of hygienics is really positive, both in mite numbers and survivorship. So so I think this is a kind of continuance of high virus loads. But if those hygienic bees had done their job and they cleared out the virus, which we did not test, or cleared out the mites rather, or the sick bees, the net at the colony level would probably be positive.
Okay, I think that's all the questions. So appreciate talking. I guess you have a little break here until 12 with the grad students. So
okay. All right.
All right.
Thank you. Yeah.
Thanks. Take care.
Yeah.
Thanks, everyone.